Hello everyone and welcome back to our channel Biodiversity and Conservation. I am Subhalakshmi Rao from Ambika Prasad Research Foundation, India, and today we are going to talk about some important experimental designs that are used in our research. So let's start. So when we say experimental design, what we mean by that? It's actually the structural or framework of the experiment, and we have actually classified the experimental design into two broad categories, that is informal and formal experimental designs. So in case of informal experimental designs, it's like the name only says informal, means we can be casual about it. Like we have some uh, less structural, or there is very less framework um, during the analysis portion. And in case of formal experimental designs, there are more precision and more control over the experiments, the procedures, and some informal experimental designs are before and after without control design, after only with control design, before and after with control design, and formal have completely randomized design and randomized block design and Latin square design and factorial designs. So these are the some of the important experimental designs, and we will go through one by one. So this is the informal experimental design before and after without control design. So here we actually have single test group, or a particular single area only, where the dependent variable is measured, and then the treatment is introduced, and again the dependent variable is measured, and after this experiment we will now study the effect of the treatment by measuring the level of experiment after treatment minus the level of experiment before the treatment. So this will give us the treatment effect. So this is sometimes difficult because with passage of time we um, have the extraneous variables that may affect the experiment process, which will make the calculation of treatment effect very difficult. Next is after only with control design. Here we have two groups or two test areas, um, that is test area and control area, and treatment is introduced only to one, that is to the test area, and control is used for matching, and the dependent variable is measured in both, and treatment is given to only test area, and then after the treatment introduction we will measure both the test area and control area and see that what is the difference is there. And here actually the basic assumption is that uh, because the two areas are identical with their behavior, then the treatment effect can be shown easily by by subtracting the uh, level of treatment activity in test area minus the level of experiment that does not have the treatment. And next is before and after with control design. So it is a slight change to the first control design. Um, Here only we have an extra control area, and in this we have test area where the treatment is introduced, and in control area treatment is not introduced. And after introduction of treatment, when we measure the difference, we can measure it through the change in the test area after treatment minus the original situation and before the introduction of treatment of the test area minus the level of change in control area without treatment minus the level out in the control area which is without treatment. So here um, this design is actually preferred um, among these three in case of informal, because it avoids the total uh, variables that are present here and may give us the accurate data. Next is completely randomized design. Here it involves two principles, that is principle of replication and principle of randomization. And here in any population, when we randomly select, that is called sample, and from that we divide them into some groups, and so basis on that we have um, two types of randomized design, and they are two group simple randomized design and random replications design. So first one, two group simple randomized design, it has actually when we divide the population into two groups, that is experimental group and control group, then we add the independent variables as treatment that are different, and then we compare the um, results. We will find some um, different hypotheses to these two, and this actually does not control the differential effects of the variables, and so we can use this method easily. And so next is random replications design. Here uh, we can take groups more than two, and first we have to take a population, then we can take here two samples, then we can divide those samples into as many groups as you want, maybe four groups each. Then we can apply two different treatments so to two major groups containing of four each minor groups, or how many groups we want to divide, because we are using here the replication technique. In this replication technique we can uh, avoid the error, and because we are repeating the treatment, and it actually serves two process, that is it provides control over the differential effects of the variables, and next is it randomizes any individual differences that are found during the conduction of the experiment. Next is randomized block design. It is an improvement over the complete randomized design, and here the main principle is that principle of local control. Here, here we divide the subjects into groups known as blocks, and within each group the subjects are distributed homogeneously, and we take some variable as a measure so that we can divide the subjects homogeneously, and uh, the number of subjects in the block would be equal to the number of treatments, and one subject in each block would randomly assign to each treatment. So here this actually helps to um, reduce the extraneous factors or the variables, and we can have a very less error. Next is Latin square design, and it is experimental design that is used in agricultural research. So um, maybe, for example, we have an experiment where we have to study the effects of 5 different varieties of fertilizers on the yield of a particular crop. So then in this case we can measure the varying fertility of soil in different blocks, and um, then we can study the effect of fertilizers on the fertility of the soil, and to do this experiment easily, Latin square design is used, and uh, here we can use two factors, fertility factor and the effect of fertilizers on the varying seeds, and this um, blocking type of design helps us to avoid all the variables, and the merit of this design is that it enables us the differences in fertility gradients in the field to be eliminated in comparison to the effects of different varieties of fertilizers on the yield of the crop. But there is one limitation also. It is that each row and each column here represents equally all fertilizer varieties, and um, there may be sometimes differences in each row and column across the field, and maybe this design is um, not field based and cannot help that much, but we can avoid the error here. So the next one is factorial designs. Factorial designs are used in experiments where effects of varying um, more than one factor are to be determined, and uh, they are especially important in several economic and social phenomena where large number of factors affect a particular problem, and there are of mainly two types: complex and simple factorial designs. In a simple factorial design, we consider the effects of two varying factors and on a particular dependent variable like height, weight, or age, or something. And the experiment is done with more than two factors. That in that case, we call it complex factorial designs. And the simple factorial design is also termed as two factor factorial design. And in this design, the extraneous variable is controlled by homogeneity, is called the control variable, and the independent variable which can be manipulated is called experimental variable. So here we are taking two treatments of experimental variable and two levels of control variable. So therefore we have four cells here, and each of the four combinations will provide us one treatment, that is experimental condition, and through this design we can study the main effects of treatments as well as the main effects of the levels that are controls. And from the complex factorial design, Here we have three or more independent variables simultaneously, and in case of three factors with one experimental variable having two treatments and two control variable, it is termed as 2 into 2 into 2 complex factorial design. And here we can determine the main effects of the experimental variable, and the researcher have to compare so many data in different cells, or like in treatment A we have 4 cells, in treatment B we have 4 cells, and we have to compare all that to get the data. And this method is used because it uh, comparatively reduces all the error. So this is all from me today. Thanks for watching.